Now, we would like to begin Dr. Tim Hunt's lecture. Dr. Takashi Toda from Hiroshima University will act as the moderator of this session. Please welcome Dr. Toda.
Thank you. This is Takashi. It is a really great honor for me to be here as a moderator for today's speaker, Dr. Tim Hunt. I just briefly introduce his profiles and careers. Dr. Hunt is from UK, United Kingdom, born in Cheshire. And he entered Career College, Cambridge, and received a PhD there. During this period, his main research focus is a control of synthesis of hemoglobin. I don't know if you have heard. This hemoglobin is a really important protein because it carries oxygen in the blood, so we need it. And following his PhD, he went to States, I think New York City, and spent several years, and back to Cambridge to take a position there. And during this time, maybe late 70s or early 80s, he frequently or regularly went to States to study biology. The place is called Marine Biological Laboratory, MBL, which is situated in Woods Hole, very famous marine laboratory for more than 100 years. So, and it was in this institute where he made a seminal, very famous discovery on the mechanism of how our cells divide. Not fallen down, divides, yeah. Cells has to divide, which is a mystery for more than 100 years. So basically he solved this fundamental problem. And this discovery led him to receive Nobel Prize in 2001, which is Physiology or Medicine. And as you know, not only he's a superb researcher, but also he's a very good speaker as well. So that you will learn a lot soon. And also, I'd like to say, he's also one who mentor for younger generation, by which now his student postdocs are becoming leader in the field all over the world. Lastly, very briefly, let me introduce why I'm here. I met Dr. Hunt several times in Japan because we have meeting in Japan. But also, I'm very fortunate because I went to London, then Imperial Cancer Research Fund, and now called Francis Crick Institute, where Tim Hunt is also there. So that's why I got to know him. In fact, I have 2 joint papers with him. That is probably one of my proudest and arguably happiest experiences in my life. So now I'm very excited about Tim's talk today. So now it's Tim. Please welcome Tim Hunt.
Okay. Ooh. Thank you, Takashi, and I'm a very lucky boy, actually, to be invited back to these HOPE meetings. I think this is my 3rd one. And as you can see here, the last one must have been in 2019, and it was in Okinawa, where the quality of the fish was a lot higher than it is here. So I'm going to tell you the story of my scientific life. And I started out, I went to my supervisor, Asher Corner, alas no longer with us, and said, what should I work on? He said, Tim, why don't you go to the library and find yourself a problem? So I went to the library and spent a week or 2 there, found a great problem to work on. I think it hasn't been solved to this day. but alas, it didn't work for me. And it didn't work for a very simple reason. I hadn't read the fine print of the materials and methods well enough, and I used the wrong kind of rat. But it was lucky I used the wrong kind of rat, because as a result, the project didn't work, and I stumbled on a much better project. And that was by going to my first scientific meeting, which wasn't very far away. It was maybe a quarter of a mile up the road in the chemistry department. And there I heard 2 talks. And as you'll see, talks have been very important for me as sort of spurs to what to do next. I always think that finding a good problem is one of the most difficult problems confronting a young scientist. You know, I mean, we can all think of problems, but it has to be a problem that your talents are suited to, and one that can provide at least measurable progress within a reasonable length of time. And a reasonable length of time is really, you know, the gap between 2 grant applications or 2 fellowship positions or something like that. Anyway, so this meeting was about hemoglobin, its structure and synthesis, and it was in honor of Max Perutz, who had recently solved the structure, the 3-dimensional structure of hemoglobin, a formidable and remarkable achievement at the time. And 2 talks made a great impression on me. One was by this guy here, Henry Borsuk, who had worked on the control of protein synthesis, and then a much younger man, Vernon Ingram, who had been the person who solved the mystery of what went wrong in sickle cell anemia. And basically, they both talked about the same thing, although you'll see the title of Borsuk's talk, Early Development of the Echinoid Egg Compared with Erythropoiesis. Well, that's quite a stretch. Echinoid egg, what does that mean? Sea urchin egg is what it means. And there is not much common between a sea urchin egg and a red blood cell, except that both of them are round. So what both these people were really working was, when you deprive young red blood cells of iron so they can't make heme, and here's the heme in myoglobin. I like this picture of John Kendrew inspecting his own so-called sausage model. You can see a very primitive structure. I mean, it's great that you know this was front page Nature News in its day. And they haven't even got the connectivity of the chains completely unambiguously assigned, which is good, I think. So this is myoglobin. There's the heme nestling in a fold in the protein. Here's hemoglobin, which has 4 chains, and you can't see the 2 that are behind. And if the cells that are making hemoglobin don't have the heme, then they stop making the globin. And there's a good apparent reason for that, which is if you make globin without the heme, it all precipitates and it causes a horrible anemia. I mean, this does happen in certain abnormal cases of abnormal hemoglobins. Okay, so what was the mechanism of the coordination? I mean, how did the cell know what the concentration of heme was? Well, Vernon Ingram had reported some experiments which he was doing. They weren't quite finished, but it was good. He talked about them nonetheless. He had evidence that what happened was that here are ribosomes reading this messenger RNA for globin, and what happened was that at this point here, the protein should have put in the heme, right? So the pocket was finally there. And if the heme wasn't there, they waited for the heme before they went on, okay? Gosh, how did they know to stop? I mean, you couldn't imagine what the mechanism might be, but nevertheless, that's what the data said. Except that when I went back and reported to my friends back in the lab, quarter of a mile away, and explained these results, we went over the methodology and we realized that this great scientist had actually completely misinterpreted his results. If anything, it was the other way around, which it made even less sense, actually. And this is the point where I can't quite reconstruct. We decided to see for ourselves, which is always a good thing to do, actually. And when I say we, actually, at that time, it was this guy, Lou Reichart, who happened to be trying to work on this project, actually, as it turns out, unsuccessfully, and he eventually became a neurobiologist, a rather distinguished neurobiologist in Stanford and later in UCSF. And I was joined a year later by Tony Hunter, and basically, who is also a very distinguished scientist. So I was lucky to be in incredibly good company as a graduate student. Very smart people. And here are the reticulocytes, the immature red blood cells. They stain with a dye that stains RNA, basically. And you can see this prep, actually, which is a photograph I took myself very many years ago. It wasn't very good. I mean, there should be more of these guys. You can see the faint outlines of the red cells here. And the preparation that it was easy to make rabbits anemic and get this blood. And Tony and I did experiments for our PhD together. And what we found was actually it was complete nonsense. There were no cues under any circumstances. We did controls that if we deliberately made cues, which we had various ways of doing, then we could detect the cues. So our methodology was all right, and these were published in quite, respectable places. So I already had I could go on with no problem. And I show this thing. This is actually a commercial thing. I think Promega. Does it say on the bottle? I can't quite see. Anyway. If you take these cells and you wash them in physiological saline, you suck off as much of the saline as you can, and then you simply add water. They burst, and you have pure cytoplasm. And this pure cytoplasm works just like the real thing. And on the right here, it shows what happens if you leave the heme out of. You have to add back some things to make it work. But basically, this rate of protein synthesis, so this is radioactivity and protein up the side here, time along the bottom. This rate of protein synthesis is pretty much the same rate that they would have done if you hadn't burst the cells open. It's amazingly good. The preparation is terrific because you can freeze it in liquid nitrogen in small aliquots. We used to use about 1 mil typically for the small ones. And that was enough to do lots of experiments with, because each incubation might be 50 microliters or something like that. It was wonderful. And so you could do an experiment any day of the week, any time of the day or night. And when you were really working hard, you could do about 3 experiments a day, which was great. It meant that the rate of progress was rapid. And in 1966, I met an American who was a hematologist, was interested in this same problem, called Irving London. And he said, what are you going to do when we fell into each other's arms, so to speak. And he said, what are you going to do when you finished your PhD? And I said, to hell with that. Why don't I come and work in your lab this summer? And so I took a sabbatical as a graduate student, and it was great fun, and I learned a lot about American science, and I liked living in New York. And I actually lived in the dorm, which was not air-conditioned. The lab was air-conditioned, so the lab was really the coolest place to be in more ways than one. Anyway, and I did go back when I'd finished my PhD and work in his lab, because I knew that I liked the people there and I liked the general thing. And this was the problem that we worked on. By this time, we had solved Lou Reichart's problem, which Lou hadn't managed to do, and somebody else had shown us the way. This wasn't our discovery at all. That when you added heme, protein synthesis kept going, and if you left the heme out, it stopped. And if you added the heme back, it started up again. So this was a reversible control system, and the question is, what was it? Well, this was an incredibly reproducible result. But if you keep on doing the same experiment over and over again, of course, you don't learn anything new, except that it works. And basically, we couldn't think of a way of getting our foot in the door. And I guess the reason for it was A, because we were pretty stupid and naive at the time, and second, because we didn't really know how the system worked at all. We didn't know enough to know what we needed to know. And so I got rather frustrated, and I had other projects that I would do after my technician had gone home for the night. So there were after dinner and weekend experiments which I did. And one of them was done with Ellie Ehrenfeld, who worked on poliovirus, because I worked out that I had a method for sequencing the genes in poliovirus using the rabbit reticulocyte if it would translate polio messenger RNA, which you get from polio-infected cytoplasm. So I asked Ellie for some polio RNA, and it didn't translate. This is a perfectly correct result. Reticulocytes don't get infected by polio. So I thought, well, there must be factors in poliovirus-infected cells that allow the translation of poliovirus, so let's get some poliovirus-infected cytoplasm and add it in, and then it'll work. But exactly the opposite happened. When you took poliovirus-infected cytoplasm, it shut off protein synthesis, whereas control cytoplasm from the same cells not infected with polio had no effect whatsoever. So there was something in this. And that something took us a long time, about 6 weeks, to identify, and it was really, really puzzling. And Nixon was bombing Cambodia at the time. It felt almost immoral to be doing experiments in the lab when such terrible things were happening in the world, which one was powerless to influence. And we used to go down to Washington I got tear gassed one time, much to everyone's displeasure. But anyway, it turned out in the end that it was double-stranded RNA, tiny amounts of double-stranded RNA. One double-stranded RNA molecule of the size of poliovirus can inhibit the synthesis by something like 10 million ribosomes. I mean, there's just no room on, it had to be a catalytic inhibition. And again, I didn't have the means for investigating that further at the time, and there was another one which did the same. So in a sense, my postdoc was extremely unsuccessful, and after about 2.5 years, Irving had by that time moved to MIT, so no longer had a lab at Einstein. I liked Einstein very much because the people there were very friendly and collaborative and helpful, and I had help not only from Ellie, but also from Jerry Hurwitz and people like that around. There were a lot of very, very good scientists there and a very nice atmosphere. And I learned a lot about Jewish customs too, because occasionally you'd go to the canteen and you had to go through different channels because of whether it was milk or not milk, and what day of the week it was and stuff. It was very interesting. And nobody, everyone went home on Friday afternoons and stuff. I mean, all this was new to me. And I hadn't realized also, this is how naive I was at the time, that, the Last Supper, which is very important in Christian theology, was actually a Jewish Seder. I mean, this was news to me at the time. So, this was an education in more ways than one. So I went back to Cambridge, and fortunately, a graduate student who I knew but had left, he was 2 years in front of me, Richard Jackson, had just recently been appointed to a lecturer. And not only that, but he and Tony had figured out how protein synthesis was initiated. It needed a special methionine transfer RNA to get it started. And that was a huge bonus. And I was very envious of the fact that they'd been able to figure this out, and I wasn't involved at all in that. And Jim Watson, in one of his books, says the good rule of science is always to find somebody cleverer than you are to work with. And it's pretty good advice, actually. And Richard was, alas, he died a couple of years ago. I'm supposed to be writing his obituary, which is very difficult. And he was great, and it was a wonderful, wonderful collaboration. Fantastic. We got on very well together. And by this time, somebody else had found that actually it was initiation. Actually, we knew this. The problem was the initiation of protein synthesis was inhibited, but Tony and Richard had figured out how that worked, so we now knew what to concentrate on, and that a reversible inhibitor was responsible for that inhibition, but nobody had any idea what the reversible inhibitor was, and we tried unsuccessfully to purify it, which would have been one way to find. We weren't very good protein chemists, unfortunately. So again, sort of matters stood still there until a graduate student, Chris Darnborough, did this experiment. We used methionine as the label in these experiments because it was cheap. In fact, we ended up making it ourselves. All you had to do was to take sodium sulfate and feed it to sulfur-starved bacterium. And we would, 50 millicuries of sodium sulfate. I used to do these at night so as not to expose too much of the lab to radioactivity, but it was cheap and easy to make. And as you can see, it used to be that these are sucrose density gradients, and I won't try to explain that. Very few of you will have done these or know what they are, but it used to be that you had to run about 1,000 of these in order to get a PhD, or so it was said. And this is a very good example of a very successful one. So we label the extract with S35, and we analyze it on a gradient. And what you see is that here, this is a single ribosome. Here are the two components of the ribosome, the small subunit, the large subunit. Together they make the full ribosome. And here are 2 ribosomes on a message, 3 ribosomes on a message. The rest have gone off the bottom in this experiment. And the point is, if you leave out the heme, what you can see is that there's a huge peak of radioactivity associated with the small subunit, as well as with the ribosomes that are making the protein. And it was very quick and easy to find out that this was, in fact, initiated tRNA binding to the small subunit. So we realized that the way that protein synthesis started was the small subunit takes this special initiated tRNA and then uses that to find the first AUG, which is the codon for methionine in the messenger RNA. And when it finds it, it locks on and then tells the other subunit to come and join. And then they start synthesizing the first peptide bond and move on down the messenger RNA. This was incredibly controversial at the time. We met incredible resistance. It was a very difficult paper to publish this because people said it should be the other way around. To transfer RNA only bound to messenger RNA, only bound to ribosomes when instructed to do so by the message. So the converse system was very counterintuitive. Anyway, so this was the sort of first success. And then the lab burned down. And this actually, bizarrely enough, was the best thing that could ever have happened because it totally destroyed the lab, as you can see. So we didn't have anything. And the result was we had a wonderful sob story to tell. I remember calling up, we desperately needed a centrifuge. And so I called up Beckman, the company that made the centrifuges that we needed at the time. And they said, oh, well, we were going to send 3 centrifuges to, I forget, Glaxo Wellcome or somebody, one of those big companies, Shell, whatever. And they wouldn't mind if they only got 2. They could wait for the 3rd one. So, the thing that was slowest to come actually was a pH meter. So within 6 weeks, we were set up with an entirely new lab with all new shiny equipment. And actually, truth to tell, quite a lot of the equipment here was very old and rusty. So we got all new equipment and new surroundings. We had to move 3 miles up the road. And we were just across a little roadway that led to the famous Laboratory of Molecular Biology. And Max Perutz was very kind to us. He said we could use his stores and have lunch in his lunchroom, which was run by his wife. And that was an amazing privilege because the place was just loaded with Nobel laureates and brilliant scientists and their students. And we could chat to them. And the other interesting thing, these are my scientific heroes, if you like. I mean, we never dreamt that we could possibly, and in fact, I still would say that we're actually not really in the same league as these great pioneers of molecular biology, all of whom I knew quite well. I mean, Fred was very modest and kind. They were all very different from one another. That was the other striking thing. They had all had their particular strengths and weaknesses. So you sort of knew that actually, there's the only thing I would say which these people had in common was that they liked things to be very simple and clear. They didn't like confusion. They were always trying to unravel confusion. And I think that's a pretty good lesson too. But then, my own contemporaries, John Gurdon was also much older than me, but somebody whose lab I liked interacting with very much, who became a good friend. We didn't think we were in the same league, but we talked to each other and made suggestions, and that's all it takes, really, to, science is so difficult. You need all the help you can get. And describing your difficulties to other people very often gives you a clue to how to proceed. Anyway, I think partly as a result of having sort of thrown away all the old confusing data, partly as a result of having all the new equipment and new surrounding stuff, we quickly solved the problems. I won't go into the details of how we did it, but it turned out that the inhibitor was a protein kinase that phosphorylated EIF2, and there were at least 2 of these things, which was what was confusing a bit. What was that double-stranded RNA? So double-stranded RNA binds to one of these things and turns it on. Heme binds to another of them and turns it off. And it turns out we missed this. I was explaining the previous thing, we should have found the unfolded protein response kinase as well, which I think was present and which we stumbled on, didn't realize what we had. And so it's quite important, and this is still a very active area of research to this day, but I haven't made any contribution since, really. So we were quite pleased with ourselves, having solved it took about 10 years to solve that problem. And we had an EMBO meeting, EMBO workshop on the control of protein synthesis, and invited all kinds of people. And one of the people I invited, I remember that first talk from Borsuk about the protein synthesis in sea urchin eggs, which is that when sea urchin eggs are fertilized, they turn on protein synthesis. So here I am, I just solved my problem, so I don't have a problem anymore. And I thought maybe a good problem to work on in the future might be sea urchin protein synthesis, but there are no sea urchins in Cambridge, obviously. We're miles from the sea. And so Tom worked in Hawaii on sea urchin protein synthesis. I invited him to the meeting, and it turned out he was a keen cyclist, which I was at the time. And he wanted to rent a bike, but you couldn't rent a bike. So I said, why don't you borrow my bike? And so we became friends on that basis. And then at the end of the meeting, he came up to me and said, Tim, how would you like to come to Woods Hole next summer and help me teach the embryology course? So I leapt at the chance because we could work on sea urchin eggs, which was a very good thing. And it turned out to be very easy to get sea urchin eggs. I don't know how the sea urchin feels about having a 12-volt alternating current put across itself, but here it is, giving rise to its eggs. You can't see, it's a pity this is not a color photo, but these are a beautiful deep purple color, and this is Arbacia punctulata, the Woods Hole sea urchin, a female. And if it was a male, it would just semen would come out. And if you mix the two together, they get fertilized, and sure enough, this is an experiment I did myself, unfertilized eggs synthesize protein at a very, very low rate. Hardly any of the ribosomes there are active. And about 5 to 10 minutes after fertilization, there's a huge increase, about a 10-fold increase in the rate. Now, unfortunately, a 10-fold increase when you're making nothing, when it's not very many ribosomes being active. So we go from sort of 0.01% of the ribosomes active to 0.1% of the ribosomes being active. So actually, this wasn't a very easy problem to thing. But I learned that if you inhibited protein synthesis, you blocked cell division. And I started looking at these cells. And this is a speeded up movie. Here is a fertilized sea urchin egg. You can tell it's fertilized because it has this thing called the fertilization envelope around it, which stops any more sperm coming in. It's very important only to get fertilized by one sperm. And if I start the movie going, you see that what these cells do, they divide every half hour, even though the temperature is only 18 degrees. I mean, this is pretty impressive. How is it possible to divide so fast? Well, they don't have to make anything new except for DNA, basically, because they're huge cells and they have to get down to regular cell size. So they start out, several thousand-fold bigger than a normal cell would be. And so to begin with, they're really specialized for cell division. So that, and then the second summer I went back there, I met Eric Rosenthal and Joan Ruderman, who were working on the control of clam protein synthesis. And that was very interesting because in clams, unlike sea urchins, which just make more of the same proteins after fertilization, clams change the proteins they make. This is a one-dimensional SDS polyacrylamide gel with labeled proteins. And I think you can see on the left is unfertilized. This protein, this big black band here, is being made after fertilization, and it really wasn't being made beforehand. This band goes away, this band comes on, that band comes on. So we proved that that was real translational control. That was an important experiment, and it was a Cell paper after only 1 month's work, because if you work on something that nobody else works on, it's sort of rather easy to find out new things that people didn't know with techniques with which you're very accomplished. Now, we had no idea what these proteins did, and we didn't have any obvious way of finding out. This is what the oocyte looks like, and this is before and after fertilization. These pictures were taken actually years apart, but they might have been taken sort of 20 minutes apart or so. And I think what you can see is what happens is a tremendous reorganization. You can't really see the chromosomes here because they're just not visible at this stage, whereas here you can just about, if you know what you're looking at, see the spindle on which is segregating these meiotic chromosomes. And the amazing thing is that this fundamental, radical transformation in the cell is brought about without needing any new protein synthesis or any new DNA synthesis, any new RNA synthesis. It simply means modifying the behavior of the components that are already present. So knowing what you know now, you would say, oh, well, the only way of doing that is either by proteolysis or probably more likely by protein phosphorylation, which is what it turned out to be. But at the time, we had no idea. At this point, fortunately, there was another seminar that I attended by this guy here, John Gerhardt, very distinguished biochemistry, who really discovered allostery, although he didn't give it that name, and the French ran away with the credit for it. But I think John was really the first. And he's very thoughtful, very nice guy. And he talked about the work he was doing at the moment, which is on frog oocyte maturation, just like the clams, but in frogs. And if this was triggered by progesterone, and this has been known for a few years before, if I add some progesterone to these oocytes, you see they're rather pretty. They're sort of yellow on one side and dark brown on the other. And I add the progesterone, and hopefully, progesterone didn't go in. Try again. I think I do. Yes, dramatic change. A white spot appears. That's the assay for maturation. Now, what's actually going on inside is exactly the same as what was happening with the clams, a total reorganization. Actually, there's a division, which you might have noticed at the end. They sort of went into a little judder. That's the first meiotic division. You can't see the products of meiosis because they're so tiny. These cells are about 1 millimeter in diameter. And what John described was experiments that a Japanese postdoc had been doing, which had identified this mysterious thing called MPF, maturation promoting factor. That word tells you they had no idea what it was, but what Mazui did was to, first of all, investigate how progesterone worked. And what he found was that progesterone brings about this dramatic change. So the nucleus dissolves and you form a spindle with chromosomes lined up. There's a spindle with blue chromosomes lined up on it. But now this is pretty slow and it needs new protein synthesis to take place, several hours. Typically. But if you suck out some of this cytoplasm in a micropipette and inject it into a fresh oocyte, this transition then occurs much more rapidly, within half an hour or so, and does not need any new protein synthesis. So whatever it is, I mean, the properties of MPF is that it's very unstable. It seems to turn itself on. And it needs new protein synthesis to reappear. And all the evidence suggested was actually, bizarrely enough, although of course this is pure cytoplasm, so you can't say it's one enzyme, it might be several, but it was the thing that triggered entry into mitosis or meiosis. And it felt to me at the time like, a really sort of delicious problem to work on, but it wasn't what I was working on at all. But I began to it was such a nice thing to think about. And these sea urchin eggs divided rapidly. Did they have MPF? And and so on and so forth. And over the years, later on, it turned out that you could find MPF was always high when the chromosomes were being segregated, always low during interphase. And it wasn't just in frogs that clams had it. That was the first one. And indeed, even humans had it. And even more remarkably, all of these different kinds of MPF would work in the frog assay. So there was no sort of species specificity to it. So it became a really pressing problem to find out what the hell it was. And, I started thinking about this. And then working on the problem of how you turned on protein synthesis, I can't really remember exactly what I was thinking of, but I did this very, very simple experiment, which was really basically another religious experiment. It was to investigate the effects of virgin birth on sea urchins. Turned out that as well as fertilizing the sea urchin eggs, you could initiate development just by adding dilute detergents or ammonium ions or calcium ionophores and things like that. And there was ancient literature, which I rather enjoyed reading about this. And I thought it was curious to see whether you initiated the same patterns of protein synthesis after real fertilization compared with these parthenogenetic methods. But what I actually found, it made no difference how you turned on protein synthesis. The patterns were the same. But I noticed something quite extraordinary, something which nobody would dreamt. In fact, if you had asked people, I think they would have said that it was impossible. And I think it's been said before, one of the best ways of winning a Nobel Prize is to actually do something that people told you was theoretically impossible. It's always a good thing, that. And so here's the protein, which I immediately called cyclin, because I was such a keen cyclist. And because it seemed appropriate. It was the most strongly synthesized one at early times. I mean, it actually represents something like between 5 and 10% of the total protein synthesis in these eggs. And then it went away, and then it came back again, and then it went away again, and we could show it always went away just before the cells divided. And it kept on doing that every time the cells divided. If you inhibited protein synthesis, of course, it didn't come back, and the cells didn't divide. But we couldn't tell, and the other amazing thing was that if you stopped the cells from entering mitosis, which you could do by inhibiting DNA synthesis, for example, then it just accumulated and never went away. So it only went away, it was only unstable in mitosis. Extraordinary. So what was it? So you can see, this is a little bit clearer. We then looked in clams, and despite the fact they'd been working on it for something like 3 or 4 years, Eric and Joan had not noticed that these two proteins went away, because they never did that really simple experiment of just adding the label and taking time samples afterwards. They were thinking about other things, the mechanism and stuff. So when we did this experiment, and this was done by a student actually, the first year, that summer in Woods Hole, in 1982, it turned out that these translationally regulated, 2 of them, which we then called cyclins A and cyclins B, went away every time the cells divided. So it was, it looked like there must be some connection between this coming and going and the coming and going of MPF, except that 2 things, really. Number one, can you really make enough of this protein in half an hour or so to catalyze something as important and radical as cell division? That seemed implausible. Number 2, there's a very poor correlation between the levels of this protein and the appearance of MPF. This stuff, by nature, goes up linearly. It has to. I mean, that's how it accumulates, right? But MPF appears quite suddenly. So, how is that possible? Well, actually, that was something that, we didn't worry about. I mean, we just got on with cloning these things to find out what they were. And also learning more about what was known about cell cycle control. Because I knew from working in protein synthesis that, it was very, you didn't really understand what you were doing until you'd made every kind of stupid mistake you could possibly make, and then correct it. And here I was going to a really very foreign field where I had never worked and didn't know what all the pitfalls were. So this had been known for several years before, which was that you could divide the cell cycle into the period when DNA is synthesized and the period when the chromosomes are segregated. And in fact, DNA is really the key to the cell cycle. I like this. Sidney Brenner said it was when he saw the model, the famous Watson and Crick model, it was a moment of absolute enlightenment, the reduction of biology to one dimension. And he pointed out this is absolutely corresponding to von Neumann's tape. Von Neumann had lectured and written about the designing cellular automata and pointed out that they needed a set of instructions to how to build themselves and also to be able to pass those instructions to the next machine so that the daughter machine would know how to build the next generation. And when you saw this, it was sort of just gave the game away. So this is key. Prepare the machinery for copying the tape, copy the tape, the DNA, then prepare for insertion into the daughters, do the insertion, share the copies between. And that has to be done with incredible accuracy. Otherwise, there's missing information in the next generation, and it won't work. So what was known about this? Not much is the answer. The best approach had been made by Lee Hartwell, who was really the pioneer of the modern movement, who had discovered that there were mutations that you could make that stopped the cell cycle in different places, implying that different places had different requirements and maybe different catalysts. And the interesting one was CDC28, which seemed to be a real regulator because it controlled budding, DNA synthesis, and the duplication of the spindle pole bodies. It's hard to imagine that could, how that would work unless it was a regulator, because they're so different in their nature. And then Paul Nurse was inspired by Lee's work and decided to use a different kind of yeast and identified cell cycle mutants in that yeast and focused on CDC2 because he found that CDC2, if you knocked it out, the cells couldn't divide. They just went on getting longer and longer and longer. But there was one mutant allele of it called WE1, or rather, actually, it was called WE2, I think, originally, where it sort of seemed to speed up. And this WE1 mutation was also very important in the thinking because it showed that you could accelerate the cell cycle and get the cells to divide to smaller than usual size. Here's the wild type length, here's the WE1 length. And CDC2 could be either made more active or less active, and it seemed that that was also likely to be a regulator. But CDC2 controlled entry into mitosis in this yeast, and CDC28 controlled the initiation of DNA synthesis. So it came as a tremendous shock when CDC28 was cloned and found to be interchangeable with CDC2. So they were the same. And you'll notice that there was no hint of proteolysis in any of this because yeast genetics can't tell you about physiology. So where did cyclins? Cyclins didn't feature in this map at all. So how cyclins got to be associated with the enzymology of the code really came back again through frogs. And Fred Locher and Jim Muller showed, in fact, it was really Mazui who discovered that you could lyse these cells simply by centrifuging them, and you could get, again, pure cytoplasm, this time undiluted cytoplasm. And that cytoplasm, actually, you could flip between interphase and mitotic states. And in mitotic state, it would assemble these beautiful spindles with here purple chromosomes lined up on these sort of greeny yellow spindles. And now if you add calcium to that, they will actually do the business of trying to segregate. And so you can have cell-free cell cycles. This is also pretty amazing. And using this assay system, Jim and Fred were able to purify MPF for the first time and show that it contained 2 subunits. And we provided antibodies which we had by then against frog cyclins to show that the larger subunit was cyclin B and the smaller subunit, in collaboration with Paul Nurse, they did this, was CDC2. So it was, once you understood, it was really, really simple. The way that it worked was that cyclin was the activating subunit for CDC2. End of story. So what had previously seemed incredibly complicated was in fact incredibly simple once you understood. The cell makes cyclin, that turns on CDC2, that phosphorylates lots of protein and you enter mitosis. The cyclin is degraded, CDC2 turns off, and you exit mitosis. And that was sort of instantly in the textbooks. And there was an amazing moment of clarification when that was revealed in as long ago as 1988. And after that, there was a tremendous amount of detail to be filled in, and it turned out that the families of cyclin-dependent kinases and cyclins was much larger. But that's a story for another day. And I think I'm already way over time, so there's no time for questions.
Oh, thank you very much. Really nice talk. So now we have a time to take questions from audience. Over there? Just middle.
Thank you very much, Dr. Tim Hunt. I'm Takato Honda from MIT. I'm so glad I could join this meeting and coming all the way from Boston. Because the last time I met you, I was in Lindau, and there was no talk by you, so I was looking forward to listening to your talk. Then
Thank you.
Yes, and all the story is in the biology textbook when I was a high school student, but when I listened to the actual story behind it is fantastic.
Well, you can see it sort of,
Yes, winding road. Yes,
a bit windy. Yeah?
And there are many, scientists are involved in the.
Absolutely,
yeah.
Absolutely. I mean, it's not something you can do alone.
Yes.
And one of the nice things about the cell cycle field was actually there were many early participants, all of whom, some worked on flies, some worked on frogs, some worked on clams, some worked on sea urchins, some worked on yeast, et cetera, et cetera. And so we were very collaborative, actually, because we didn't exactly step on each other's toes. And I think that was a huge factor in the rapid development, early development of the field. Once we figured out the basic idea, it was very quick to go on from there. But the basic idea was, one of the things I didn't say was how stupid we should have got there much sooner, I think. But we were hindered by our lack of understanding of protein kinases. We had the example of cyclic AMP-dependent protein kinase, which has a repressive inhibitor, which is relieved by its activator. The idea that you could turn on a protein kinase by doing something, you have an activating subunit rather than a repressing. I mean, isn't that, why has nobody even sort of suggested that as a possibility? So why not? I can't emphasize enough really how our preconceptions limit our understanding. On the other hand, it's a strength, I think, of scientific method, because then you're confronted by a result which is only interpretable one way, and this makes it very, very strong, actually. So I think experiments are the thing. Theory can blind you. Experiments can reveal the truth. That's not to say that theory is unimportant, though.
So, I actually haven't asked you a question yet. So, my question is about time management and
time management. I'm very bad at that.
No. And also some, how to be independent. So this question is, I'm asking this because your major discovery is mostly found in Woods Hole.
Yes.
Right? So, but far from your home institute. So it means you had also the time in spring and autumn and winter. So
yeah.
Then what I wondered what you were doing the rest of the time.
Okay.
Because I'm asking this, as I told you the other day, I'm also going to join those independent research program this summer at Woods Hole. So then that project is completely independent from my main project at MIT. So I just, raise an idea and submit a proposal. Then I'm very excited, but also I try to worrying about my main project as well in the lesson. So I just wondered, how did you manage?
Yeah, no, that's a good question. Worrying about the main. Well, I think the answer is don't worry. Don't worry at all. You're more likely to get stimulation by going to Woods Hole than you are to lose. You just keep doing the same old thing at home. And going someplace else is tremendously rewarding because you talk to people, you learn, if you do it right. I went to a lot of lectures and seminars. So, when it was constant stimulation, a real intellectual ferment, it was wonderful. I learned so much cell biology, which I would never, ever have learned if I stayed home by going to Woods Hole. And I also learned a sort of lot about the way of doing research. I created over the years a sort of little home away from home. The first year I went there actually was a complete disaster scientifically because I didn't have the kind of equipment I needed. So the next year I went with a suitcase full of all the really essential little things you need in a lab so that I could get to work immediately. It was interesting. People said that they didn't want their graduate students to go on these courses because it would take them away from their project. But I sent my graduate students, the ones I thought would benefit on these courses, and it was amazing the effect that it had on them because they would learn new stuff. They would learn their place in the universe of international science too, because they would discover that they were no cleverer or dumber than the other students from all over the world. They would also learn that you could learn how to clone and sequence in a week, actually, if you really worked at it hard. These things weren't ridiculously esoteric. You could just do it. And so these were all fantastically useful lessons. And I think, having something that automatically sort of shakes you out of a rut is awfully good, actually. I think you can then sort of stand back. It's a little bit like putting a manuscript in a drawer and coming back to it months afterwards. You suddenly realize all the stupid things you said and how they could be put better and where your mistakes were. It was lucky. I would say one thing. I didn't have a family at that time. I was completely... And the graduate students who were left behind, actually, I always wondered, they never made much progress while I was away, which surprised me because, they were totally in control of their own fates then. And what we did in between, that was also part of the question, was, so the next summer, the first job was to confirm that cycling really went away, and I began to think it was all a dream. But it did go away. It kept on going away. And then the problem was how to clone it, and I had no experience in that, and that took us rather longer than it should have done because I didn't know how to do it. So we had to sort of teach it was stupid not to go on a cloning course, actually. I don't know why we didn't do that. But anyway, we managed it and invented. I had some good fun along the way, but I never talked about that, actually, but it was good. I had two graduate students, John Pines and Jeremy Minchell, and they were both, in their different ways, absolutely wonderful. And it was another example, actually, of where, I've been happiest. I mentioned Richard Jackson. He was a great friend and collaborator. I've always felt happiest when collaborating with just a very, very small number of people. Best at best, maybe one or two at most. I'm not the kind of person who can run a great big team. I don't know how to do that. So it depends critically on having really good people to work with.
Thank you very much for your kind advice. And
Thank you.
Yeah, I should really appreciate my current advisor who, generously send me, giving me a shot to do some independent project. And I wish I could be like a scientist like you who can send a graduate student. Can I? Oh, maybe.
Okay.
Yeah.
Can I go to the next question? People. Yeah.
Thank you so much for your impressive talk. I'm
come up
trying.
And see if I can. I always like to sort of see the whites of the person's eyes. It turns out to be easier. And also, I'm a bit deaf.
Okay. So outside the body, the iron is oxidized into iron oxide, and it become fragile, become rust, and it is very difficult to deoxidize the iron again.
Oh no, the iron in the red cells. There's an enzyme there called methemoglobin reductase, which does the trick.
Which prevent the oxidation of blood.
No, the iron in heme is oxidized. It's in a 3+ state, right?
Yes.
And when it gets into the protein, it's still in the 3+ state. And then there's an enzyme, I forget what cofactors it uses, but it reduces the iron to 2+. And it's only the 2+ state that can carry the oxygen.
Which is stable enough not to inducing any reactive oxygen species or inducing cell
Well,
death.
it probably does, you know. I mean, these cells last for about 3-4 months or something like that, and then they die and the protein gradually gets attacked by things. It can't. The thing is, these red cells have no nucleus, they have no ribosomes, they have no mitochondria, they can't repair themselves. So they have some protective systems, but they're doomed to die quite quickly compared to most cells in your body.
And your red blood cells are produced from bone marrow.
From bone
but
marrow, mostly,
there
yes.
are cell aging where bone marrow cannot produce no more red blood cells.
Well, if you can't produce more red blood cells, you die.
Right, so it cannot stop producing it.
Well, you go on producing them throughout your life as long as you're alive.
So why I cannot live forever?
Well, that's a good question.
Like cell aging.
I think the reason actually you can't live forever is quite simple. That it's like buildings, right? It's much cheaper to knock down an old building and build a new one than it is to keep an old building in good repair. That's very complicated. And some things are virtually impossible to repair, like knees, for example. Turns out, from the atmospheric weapons testing that was carried out in the 1950s, I have radioactive carbon in my knees, which is pretty amazing because it was put there in 1955 or 56 or 57 or something like that. And it's still there because it really doesn't turn over. And if something goes wrong, that lack of turnover also means you can't make new stuff. So there are some things you can repair and other things you can't repair. But the best way of making, it's much easier to make a new human being by fertilizing a human egg than it is to keep an old one going. So I'm about to die.
I wish not. So thank you so much.
Thank you.
So there's a question here.
Thank you, Professor. I asked you yesterday about the sea urchin, but I'm not going to ask you that question. But since you mentioned, I will ask you probably another stupid question now, since I'm not someone that's doing cell biology. So since you mentioned about oyster, progesterone, MPF, etc., I'm just kind of like interested if you know, and also the fertilization, I kind of like want to know if you know something to do with the sex ratio related to this essay, probably in the past, if you know something about it.
The sex ratio?
Oyster.
Oyster?
Yeah.
The oyster.
The reason I asked that because you know that oyster changes
I know
sex,
nothing
so
about oysters except they're good to eat.
Okay, yeah. So, yeah, okay.
I'm sorry.
I know, it's only like a very stupid question. I just thought that because you mentioned about, the relation of progesterone, MPF, and also it happens in oyster.
Well, I'm sure it does, but I don't know what the hormones are that control oyster development. I mean, this is one of the problems with sea urchins was always sexing them. There is absolutely no difference between the look of a female and a male sea urchin because rather like fish, they just put their gametes into the seawater and they don't have to meet or kiss or anything, right? Yeah. Although it turns out the fish get together to mate. I mean, I once saw an amazing movie of what happens it was Drake's Bay, and the sea just becomes white with fish semen and the eggs, so you never know if you're a fish, you never know who your dad was.
Very sad for them.
Or your mum, for that matter.
Okay, Tim.
Thank you.
That will the maybe last question. Someone? Yeah.
Thank you for your talk. I'm not a biologist, but I spent 2 years doing biology in my master's. And I wanted, it's maybe out of the scope of the specific lecture, but I noticed that now many biologists just buy kits, like pre-prepared kits, and I did it as well. I needed to do immunogod labeling, and I did that. And it really bothered me that I have no idea
what's in the kit.
I just, follow instructions. I don't know the concentration. I don't know pH. I don't know anything.
Yeah.
And I saw that all my friends, all my biology friends that I worked with and still, look at the halls from time to time, they do it. And I just want to, you had so many controls. You showed us so many controls here. I just want to know, what do you feel about it? How can you make fundamental research when you don't know what you're actually doing?
Yeah, no, that's a good question. I don't really approve of kits, except that I was involved in making a kit once. It was great fun. We made a cell-free translation kit for assaying messenger RNA together with Amersham. And, we had huge debates about what kind of tube to provide and whether to give people water and stuff like that. But an interesting story is I had a graduate student who came with me when I moved from Cambridge to ICRF in London. And her reaction stopped working. And she thought it must be that, she'd made up one of the reagents wrong. And it took her the longest time to discover that actually the problem was not her work, but the work of the company, which was a very reputable company who supplied the key enzyme. Up to that point, we made the enzyme ourselves. But I thought, we're so rich now, we don't have to make the enzyme ourselves. We'll just buy it from New England Biolabs, which is a really good company. But actually this thing, it's clear somebody just hadn't stirred up the solution properly. So all they sold us was bovine serum albumin with a tiny trace of T7 RNA polymerase. And that's sort of a very instructive example, it's caveat emptor. And so if you make the stuff yourself, it's good. If you buy a kit, you're dependent upon all kinds of other people. And as you say, you don't really know what's in it, which means you're already, I agree, you're sort of treading on thin ice, I think.
That was my reason to leave biology, by the way, because anyways, there are so many dependent...
And people, the other funny thing was that, people trusted these kits more than they trusted themselves.
Correct.
Which was stupid, actually. I guess, I sort of felt a bit the same way myself. Because I suppose, when you're in that business, this was something I also learned from helping Amersham develop the kit. To begin with, it was kind of interesting. We said there was a lot you had to think about and what you had to provide and how to do it and da-da-da-da. But after a bit, I realized that, the business of making commercial reagents was terribly boring, actually, because you didn't learn anything new. You learned sort of tiny, tiny details. And it was sort of not very satisfying in the end. But there were other people who obviously enjoyed it and were good at it. And, companies like New England Biolabs have made a tremendous contribution to advances in the field by seeking out new reagents. And exotic reagents can be very, very useful in providing tremendous reliability and documentation and so on and so forth like that. But after all, there was a time before my time when people had to make their own ATP. So you went to the slaughterhouse and you got a dead horse. And, and we didn't think anything was wrong with buying ATP. But the previous generation would probably have said, they don't even make their own ATP. So I'm not quite sure where...
So you're saying that a good balance should be made?
I think, kits really do speed things up when they work. But you're right, they sort of remove some of the... It's like buying I think it's better to make a cake with flour and sugar and eggs than to buy a thing in a packet and just add water, myself, because then you sort of know what you're dealing with and you have more control over the variables. Although it's true that the Betty Crocker cake mix will actually make you a pretty bloody good cake if you follow the instructions on the packet, And if it doesn't, you can sue them or get a new one.
Right.
I don't know. I think you've got to accept the kits, though, for lots of things, I'm afraid. But it's better if you understand what's in them than you don't, because you may end up, with some incompatibility down the line if you don't really know what's there.
Thank you.
Thank you. Tim, please carefully come back. Okay, now this is all in this morning session. Thank you, Tim.
Thank you very much, Dr. Hunt and Dr. Toda. Let us show our appreciation with a round of applause.